proteins, fats, and sugars on the table, so to speak, and we've discussed how enzymes speed up biochemical reactions so they can take place at what I call the speed of life. But how does life safely use and manage all the energy of those reactions? It's an incredible story of energy storage and transformation that centers on a molecule known as ATP. Now, amazingly, we make and break down our weight in ATP every day. To understand how it's possible to do this, let's start by considering the sugar in a marshmallow. There are basically two ways the sugar in that marshmallow can be converted to energy. First, think about what happens if you accidentally set fire to a marshmallow you were trying to toast. The burning sugar of marshmallows makes for some pretty impressive flames. That's oxidation, and heat is released in the process. Now compare that with what happens when you eat an uncooked marshmallow. Same marshmallow, sugar again converts to energy, and it's still oxidation, but you do not burst into flames or start breathing fire. What's the difference? What cells do during their oxidation of glucose happens in many steps instead of one big one. The energy released in each step is correspondingly lower, so no fire results. What's remarkable is how much of the small amounts of energy released in glucose oxidation in cells is actually recaptured as biochemical energy. Essentially, one form of chemical energy, glucose, is converted to another form of chemical energy, ATP, with relatively little loss as heat. Now think about how different this is from beginner chemistry shows for the public, where the stars of the show are merely setting things on fire and blowing stuff up. In cells, by contrast, the complete oxidation of glucose requires over 30 individual chemical reactions, and the ultimate product of these reactions is another high-energy molecule, ATP. Going to all that trouble is worthwhile because ATP works like a battery, storing energy to be used instantaneously by cells. The fully charged battery of ATP can instantaneously discharge its energy and become ADP. The spent battery of ADP can be recharged then back to ATP. ATP is a triphosphate, and ADP is a diphosphate. So all we're doing is removing and adding a phosphate. This can happen over and over again when using energy from food. Creating the gigantic amount of ATP energy the body needs each day requires heavy-duty biochemistry. That's what we're going to spend most of this lecture discussing: energy transformations in biochemical reactions. Okay, so our cells get energy by oxidizing food and capturing the released energy in the bonds of ATP. Why do we need so much ATP? Cells must build and maintain complex molecules and structures. These activities require energy, whether we're stringing together amino acids to make proteins or carrying out the many reactions that build cholesterol for membranes. Moreover, all cells have two kinds of activities going on simultaneously: breaking down of food to obtain the necessary building blocks and energy, we call that catabolism, and synthetic pathways that use the building blocks and energy from catabolism to create the molecules the cell needs, we call that anabolism. You can imagine this as a construction of a house going on at the same time as demolition of a nearby house containing materials that you need to build the new house. Everything you need to build the house has to come from taking apart something else, so you must constantly be disassembling things so you can make new things from the pieces. Demolish a barn so you'll have boards to build doors with. Take apart a greenhouse to get glass for your windows. A fundamental question that arises for any given reaction is, are we building up or breaking down? Some reactions are used for catabolism in one direction and anabolism in the other. That now poses an important question. What rules govern whether a reaction goes forwards or backwards or not at all? And how can organisms manipulate conditions to make a reaction go a particular way in response to changing needs? The answers to these questions come from the subject of thermodynamics. You may remember from the lecture about the enzymes that Gibbs free energy, that we call G, is a measure of the amount of energy available to do useful work in a process. In biochemical reactions, the work being done is making or breaking of chemical bonds. As biomolecules react with each other, there is a change in the available Gibbs free energy. The size of that free energy change is called delta G. If energy inputs are required for a reaction, then delta G is said to be positive. If delta G is negative, then the reaction gives away excess energy. Let's imagine a reaction where the free energy G is lower for the final product than for the starting molecule. We describe this as moving downhill, energetically speaking. In everyday terms, it is easy to envision that a ball at the top of the hill will roll down to the bottom. The higher the hill, the steeper the slope, and the greater the likelihood of balls rolling down to the bottom. That's why we care how big the difference is between the initial free energy and the final free energy. Delta G, that difference, is the change in Gibbs free energy. And because the product is at a lower energy level than the starting molecule, energy is given off in the reaction. Mathematically, we gave away energy, so the Gibbs free energy change, or delta G, is negative. Conversely, to change a molecule that has low free energy to a molecule that has high free energy requires input of energy, just as pushing an object from the bottom of the hill to the top requires added energy. Since the object is gaining energy in this process, the change in Gibbs free energy is positive and the value of delta G is positive. But how do we tell for a given reaction what its delta G is? Let's consider a simple reaction. A goes to B. When we think of a reaction going from A to B, we might imagine it only goes one way. But it turns out that cellular reactions are never that simple. Instead, it's far more common for a reaction to go mostly one way, but also somewhat in the other direction at the same time. Because of this, it's rare that a reaction goes all the way until A is converted all the way to B. Instead, there will always be, to a small extent, conversion of B back to A. The outcome of the reaction is a matter of how much of A is converted to B and vice versa. Which brings us to a second consideration, and that is the relative concentrations of A and B we start with. Suppose we start with only A. The reaction converting A to B would proceed, making more and more molecules of B. What if we started with only B and no A? Since the reaction is reversible, B would start going to A. In neither case, though, would the reactions get completely converted into A or into B. They will stop at a point somewhere in between. In fact, when we measure the ratio of B over A, at the point where they stop, the value of that ratio will always be the same, no matter what amount of A or B we started with. This midpoint in between, where the ratio of B over A stops changing, is called equilibrium. The ratio of B over A at that point is referred to as the equilibrium constant. There is one such equilibrium constant for every reaction, and here's a surprise. The equilibrium is almost never when the two concentrations are equal. The equilibrium for one reaction might be at 60% B and 40% A. Another reaction where C goes to D might be at equilibrium at 20% D and 80% C. Each reaction has its own equilibrium constant. It doesn't matter what the starting concentrations of the molecules in the reaction are. At equilibrium, the ratio of B over A or D over C will always have the same value of the equilibrium constant specific to that particular reaction. Let's say that for the A to B reaction, the equilibrium constant is 70% B and 30% A. What will happen if I start with equal amounts of B and A, say 50-50? In that case, the reaction will go forwards until it reaches 70% B and 30% A, because reactions always move towards equilibrium. If I start with 90% B and 10% A, the reaction will go backwards until I reach 70-30 again. So the equilibrium constant and the amounts of A and B I start with determine the direction the reaction goes. Okay, reactions move toward equilibrium. How do we know if a reaction will go forwards, backwards, or already be in equilibrium? Well, delta G gives us an easy way to predict that. When a reaction is at equilibrium, its delta G value will be equal to 0. When the delta G is negative, energy is released and the reaction moves forwards. If the delta G is positive, their energy would have to be added for the forward reaction, so it moves backwards. Delta G tells us a lot about reactions. So how do we determine the value of delta G? Well, this turns out to be related to the equilibrium constant. Let's look at an example. To do that, I'm going to go through a bit of math, but you don't have to understand the math to get the take-home message. Bear with me. Let's start with a 1 liter solution of A and B, 50% of each. Let's have them at what biochemists call standard conditions for a reaction. This involves a temperature of 25 degrees centigrade and an atmospheric pressure of 1 atmosphere. For temperature, scientists use the Kelvin temperature scale, which puts 0 degree Kelvin as absolute zero. So 25 degrees centigrade is 298 Kelvin. We know that every reaction is going to move towards equilibrium. We simply need to determine if there is energy released by the reaction, that is negative delta G going downhill, or absorbed by the reaction, positive delta G going uphill. First, we'll measure the value of that energy change. Eventually, the reaction will reach equilibrium and the energy will not change. At equilibrium, we'll measure the concentration of B and A, since their ratio will give us the equilibrium constant. So we have 2 conditions and they correspond to 2 energies. There's a starting condition not at equilibrium, giving off or absorbing energy, and there's an ending condition at equilibrium with no energy change. In essence, the starting condition must be neutralized by the forces making the ending condition. In other words, the starting condition gives off energy, which is negative delta G, or absorbs energy, so delta G is positive. The forces giving rise to equilibrium must be countering that negative or positive energy by creating an opposite energy of an equal amount with the opposite sign. Now, in the beginning, the countering energy is equal to 0 at standard conditions. The absence of countering energy is what allows the reaction to move towards equilibrium. So this is a big simplifier for us. Delta G at start equals initial energy plus 0, the countering energy at standard conditions. Since the reaction is not at equilibrium, it moves towards it. Then it reaches equilibrium, and when that happens, delta G at equilibrium equals 0, which is equal to the initial energy plus the countering energy at equilibrium. Thus, the countering energy at equilibrium must be opposite in charge and equal in magnitude to the initial energy to bring the delta G to 0. The initial energy was the energy released at the beginning under standard conditions. Biochemists call the initial energy release the change in standard Gibbs free energy and give it the symbol of delta G naught prime. Now, the naught indicates the measurement was done at pH 7, as would exist in a cell, and the prime is used to distinguish it from delta G. Each reaction has its own distinctive standard Gibbs free energy change, and it remains a constant for that reaction. The countering energy at equilibrium is the negative or opposite in sign of the initial energy. For instance, if the initial energy was 4 kilojoules per mole, then the countering energy at equilibrium must be minus 4 kilojoules per mole, since 4 minus 4 equals 0 equals delta G at equilibrium. There is an equation that captures all of this. The idea here is that delta G changes depending on the concentrations of product B and reactant A. Here's how we write the equation. Delta G equals delta G naught prime plus R times T times the natural log of B over A. And since there can be more than one product created by more than one reactant, we simplify B over A to products over reactants. Let's think about what this means in words. First, delta G, the change in Gibbs free energy, is the actual change in free energy for a reaction, which tells us the direction of the reaction. A negative value means the reaction goes forwards. A positive value means more energy would be needed, so the reaction on its own must go backwards. Reactions at equilibrium have delta G equals 0 and have no further energy change at all. Second, the delta G under standard conditions is always the same for a given reaction, so it's a constant in the calculation of actual delta G. Third, the only variables in the equation that cells can change are the concentrations of reactants and products. Fourth, R is simply a constant as well, and T, for the fifth point, is the temperature in Kelvin. Last and sixth, the term natural log of B over A is positive when B is greater than A, it's 0 when B equals A, and it's negative when B is less than A. So the ratio of the product to the reactant is the main term influencing the whole equation. Thus, the countering energy is positive if B is greater than A, and negative if B is less than A. And as the concentration of B increases, the natural log of B over A term gets more positive. The more positive the natural log term is, the more positive the overall delta G becomes. But if more and more energy is required, that means the reaction will tend to move backwards. Now that makes intuitive sense. Increasing the amount of product will move reactions backwards. Conversely, increasing reactant A will eventually make B over A smaller than 1. Now that means that the natural log term will become negative. It's giving away energy, and the reaction will be favored to move forwards. Now cells exploit these principles all the time, altering the concentration of reactants and products to favor a reaction going one way or the other. So again, the key idea is delta G changes depending on the concentration of products and reactants. Very important point. More reactants favors forward reactions, more products favors backwards reactions. That's pretty simple. In turn, it means that if a cell manipulates the concentration of either products or reactants, the delta G will change. It can be changed from negative to positive or vice versa simply by altering the concentration of products and reactants. Why is that so important? Well, drum roll please. The changes in delta G are the secret to how cells control the direction of reactions. It is, in fact, the only control that cells can exert over the direction of a reaction. O delta G, the change in Gibbs free energy, can tell us if a product will advance. 'Cause if the value's negative, it translates that reverse reactions haven't got a chance. But when the sign is positive, it's opposite. And then the backwards happens all the time. A constant is the standard Gibbs free energy. So don't forget about the delta G naught prime. Okay, we've got Gibbs free energy and how it changes under our belts. Now let's take a real-world look at how our cells change the direction of reactions in response to changing needs during exercise. ATP, as we saw earlier, serves as a key source of energy for cells. One of the uses of ATP energy is in muscular contraction. The contraction of muscles underlies such involuntary actions as the movement of materials in our digestive system, the beating of our hearts, as well as the voluntary movements like dancing and running. When muscles work, ATP is broken down, creating ADP, and the energy released in the process fuels this contraction. To replenish ATP, ADP has to have a phosphate added on again. Now, keeping ATP concentrations high is a no-brainer for muscles. A reliable ATP supply is so important that muscle cells have an emergency backup system based on a molecule created mostly in the liver and kidneys called creatine. High-performance athletes may be familiar with creatine as a supplement taken with hopes of improving performance. That idea is based on the simple equation that we're getting ready to talk about. Remember how muscles use myoglobin as a backup source of oxygen? Myoglobin held onto oxygen tightly and released it when cellular oxygen levels dropped to a very low level. It turns out that for ATP energy, a phosphorylated form of creatine called creatine phosphate acts in a very similar backup role, but it supplies energy, not oxygen. Making creatine phosphate requires energy from ATP in the first place, and this is done when ATP supplies are abundant. The phosphate added to creatine can then be transferred to ADP to make ATP when levels of ATP drop. The equation that we have for this is ATP plus creatine goes to ADP plus creatine phosphate. The change in Gibbs free energy for this reaction under standard laboratory conditions has a numerical value of positive 12.6. So the positive change in standard Gibbs free energy for this reaction means it would be unfavorable under standard conditions and would go backwards if we started with equal amounts of everything. However, and fortunately, resting muscles don't have materials present at standard conditions. The physiological conditions that muscles have favor a forward reaction with a negative delta G. How do they do this? Well, let's take an example. When you're at rest eating pizza, for example, levels of ATP in your muscles are relatively high, and levels of ADP are relatively low, because you aren't exercising and using that ATP. The low concentration of products, in this case ADP and creatine phosphate, in the numerator, and high concentration of reactants, ATP and creatine, in the denominator, means that the value of products over reactants is considerably less than 1. The logarithm of a number less than 1 is always negative, and the more ATP and less ADP there is, the more negative this number will be. So even though the reaction has a positive change in Gibbs free energy at standard conditions, a large negative value for the log of products over reactants will bring about a negative value for the change in overall Gibbs free energy. And because of the negative Gibbs delta G, the reaction will move forwards, resulting in synthesis of creatine phosphate. So at rest, the body is making creatine phosphate, exactly what we want, since it is our backup ATP source. Under high ATP, while resting, the reaction is driven to the right and creatine phosphate is made, as I said, very important. If you then go for a sprint, the first thing that happens is your muscles use ATP, which gets converted to ADP. Levels of ADP rise as a result. This can be problematic for performance if you didn't have a backup, but fortunately you do in the form of creatine phosphate, made while you were at rest. High levels of creatine phosphate and ADP now means that products is large relative to reactants, and under these conditions, delta G is positive. It drives the reaction back to the left, creating more ATP and creatine. Now, while running, ATP is low, and the reaction is favored to move left to make more ATP. ATP, of course, is broken down to fuel the muscles as soon as it is made, making even more ADP, and that continues to push the reaction to the left. So the reaction will strongly favor the formation of ATP at precisely the times when the muscles badly need it. Later, after the run is over and you go back to resting and eating more pizza, ATP is not being used as rapidly, so its concentration on the left increases. But so does the concentration of creatine phosphate on the right, both going back to previous levels. Thus, changing levels of the reactants and products determine in which direction the reaction runs. Leftwards to make ATP when exercising, rightwards to make creatine phosphate when resting. In short, cells control reactions by manipulating concentration of reactants and products. The creatine phosphate reaction is actually an unusual way of making ATP. It is unique to muscles because it provides instant backup energy that muscles need, but it is limited in how much it can supply. When you get tired from exercise, it is because you start exhausting your immediate energy sources from creatine phosphate. Other energy sources such as fats, carbs, and proteins are better energy sources for cells because it is easier to make and store more of them. But they come with other considerations, such as how fast and how easily they can be converted to ATP. That, as we learned earlier, comes from oxidation of our food that releases energy. So let's now look more closely at oxidation. Chemically, oxidation originally got its name from reactions where oxygen was added. Reduction was reactions where oxygen was removed. But that masks what is actually happening in cells during oxidation-reduction reactions. In oxidation reactions in cells, electrons are lost by an atom or molecule. Molecules that lose electrons are said to be oxidized. Molecules that accept electrons are said to be reduced. Cells use electron carriers instead of giving electrons directly to oxygen, as would happen if you burned a substance in air. So the electrons go to carriers, and only later do the carriers give the electrons to oxygen. The most important carrier molecules ready to accept electrons are NAD+ and NADP+. These electron acceptors with the plus sign are in their oxidized states. Their reduced counterparts, the carriers when fully loaded with electrons, are known as NADH and NADPH respectively. Now, for any reaction, the total number of electrons remains the same. Anytime a molecule is oxidized by losing its electrons, there has to be another that is reduced by gaining those electrons. This close coupling of reduction with oxidation is why such reactions are sometimes called redox reactions. In cells, electron carriers are the intermediaries. And just as for all chemical reactions, transfer of electrons is accompanied by a release of energy. This release of energy, when electrons are transferred, can be used ultimately to create the high-energy phosphate bonds in ATP. Similar high-energy phosphate bonds can be used to store energy in other related nucleotide molecules like GTP, CTP, and UTP. The TP at the end of their name stands for triphosphate. And yes, these are the same molecules that are also used to make RNA, which we will explore much later in the course. Again, the common theme in all four molecules is that each has three phosphates linked together. Phosphate groups, as we saw in earlier lectures, have negative charges, so they repel each other. Joining them together requires energy, which is released when the bond is broken. So breaking the bond between two of the phosphate groups in ATP to make ADP plus a phosphate is energetically favored. This reaction is called hydrolysis because water is involved in breaking the bond. Breaking the ATP bond releases the energy put in to join the phosphates together in the first place. The energy-releasing hydrolysis of ATP to ADP is coupled by the cell to reactions requiring an input of energy. This lets ATP breakdown supply energy where it's needed. The way alcohol is metabolized is an example of oxidation. If we consume a beer, our liver converts ethanol in it to acetaldehyde. In this reaction, we say ethanol gets oxidized because it loses 2 electrons. It also loses 2 protons. The electrons and a proton go to NAD+, which starts oxidized, but we say gets reduced to become NADH. Ethanol at the start was the more reduced molecule, and acetaldehyde at the end is now the more oxidized molecule. NADH and NAD+ are continually toggling back and forth between the oxidized state of NAD+ and the reduced states of NADH in cellular reduction-oxidation reactions. The same sort of toggle goes on between oxidized NADP+ and reduced NADPH. NAD+ is a crucial electron acceptor for catabolic oxidative reactions during respiration. NADPH is a common electron donor in anabolic reactions used to build molecules. Thanks to electron carriers, electrons are contained and oxidative energy ultimately gets directed to the mitochondria to make ATP. Whether a cellular reaction goes forwards or backwards is governed by thermodynamic laws that cannot be violated. Like us, our cells have to be law-abiding citizens. A reaction cannot, for example, go forwards if its overall delta G value is positive. Cells perform the remarkable task of making all that ATP we've been talking about within the statutes defined by the Gibbs free energy equation. Fortunately, altering concentrations of reactants and products is all that's needed to move a reaction in a desired direction. And cells are very good at changing the concentration of reactants and products. So you've just worked through some of the most important foundational concepts in biochemistry, ideas that we will use to understand the forward and backward direction of entire pathways of biochemical reactions. We're ready to explore metabolism, and we'll start with sugar.